The next step promised to be even more difficult. Learning that New York is halfway between Washington, D.C. and Boston is better than merely knowing it is somewhere in the United States. But when one must set out on foot from Washington to Boston, looking yard by yard for a sign that reads, "Welcome to New York," the clue may seem not so helpful after all. 1 million base pairs might be close by the standards of linkage analysis, but it is a very long way by the standards of gene cloners who analyze regions one base pair at a time. To go the distance from the two RFLPs nearest the CF gene, Sue teamed up with Francis Collins, who was then at the University of Michigan, and would later succeed me as director of the Human Genome Project. Collins had developed jumping techniques to facilitate the cloning of a gene between a pair of known RFLPs. But he was under no more illusion than Sue about the magnitude of the problems facing them. After 2 years of work, they managed to localize the CF gene to a 280,000 base pair segment of DNA, within which they found the sequence of a gene known to play an important role in human sweat glands, which are dysfunctional in cystic fibrosis patients. It seemed the complete CF gene might finally have been corralled. The only way to be sure they had got it right was to sequence the cDNA and search for the disease-causing mutations. Given a region of 6,500 base pairs long, this was quite a challenge in 1989. And it had to be done twice, once using DNA from a CF patient and once with DNA from a healthy individual. The result, however, was clear-cut. The patient's DNA was missing a stretch of 3 base pairs, resulting in the absence of just one amino acid in the protein. This one mutation accounts for about 70% of CF cases, but over 1,000 others found in the CF gene also cause the disease. This multiplicity of harmful variants has greatly complicated the task of DNA-based diagnosis. Let us now turn to Nancy Wexler, David Hausman, Jim Gusella, and their colleagues, whom we left back in 1983 at the triumphant moment when a particular RFLP, G8, had been linked to the gene for Huntington disease. If it seemed up until then that they had enjoyed more than their collective share of good luck in locating the HD gene with astonishing speed, the gods were soon to redress the imbalance. Finding the gene had taken a mere 3 years. Isolating it for detailed analysis would take 10 years and an international team of 150 scientists. In this case, the region where the gene had been localized was 4 million base pairs long. The Huntington disease geneticists worked hard to narrow that window. But genetic mapping gets more difficult as the genetic distance gets smaller. And finally, these efforts were rewarded only with ambiguous data. Imagine the foot journey from Washington to Boston in search of New York. Now imagine arriving at an intersection in Philadelphia to find a signpost indicating New York in both directions. Well, giving up on the contradictory linkage analysis, the Huntington gene hunters devised an alternative strategy, focusing on the region that was most similar among Huntington disease patients. This approach eventually reduced the region to only 500,000 base pairs, and the time had come to turn to gene cloning techniques. The first results were disappointing. They found 3 genes in the right-hand half of the region, but none showed any abnormalities in patients with Huntington. Undaunted, they explored the left-hand side and found a single gene with a prosaic name, IT15. Finally, after 10 years and many losing lottery tickets, luck had begun to smile on them once more. The gene contained a short sequence, CAG, that repeated over and over again, like the short tandem repeats, STRs, used in DNA fingerprinting. It turned out that unaffected people have fewer than 35 CAGs, while people with more than 40 will develop Huntington as adults. In the rare instance of more than 60, a severe form of Huntington develops before the age of 20. CAG is the genetic code for the amino acid glutamine. So each of the CAG repeats adds an extra glutamine to the protein. In the case of Huntington sufferers, the protein coded by the HD gene, the rather difficult to say Huntingtin, contains extra glutamines. This difference likely affects the behavior of the protein and brain cells, probably by causing molecules to stick together in gluey lumps within the cell, somehow causing its death. It had been a tremendous effort by all the laboratories in the Hereditary Disease Foundation's team, and in recognition that it was truly a collaboration, the only name appearing as the author of the article was that of the Huntington Disease Collaborative Research Group. The same strange type of mutation, repeats of the 3 base pair sequence, had already been implicated in 3 other disorders. Remarkably, all of them also neurological diseases. We now know of 14 of these trinucleotide repeat disorders. But we still are no closer to understanding why brain cells are so susceptible to this kind of mutation. It may be depressing to know that despite the substantial time it has taken to hunt down their respective genes, these disorders, Huntington, Duchenne, and cystic fibrosis, are, by the standards of geneticists, simple. They are caused by mutations in a single gene and not much affected by environment. If you have the 3 base pair deletion in both your cystic fibrosis genes, or more than 40 CAG repeats in one of your Huntington disease genes, you will develop those disorders, no matter where you live or what you eat or drink. There is a large number of single-gene disorders. The current genetic disease database lists several thousand, but the majority are extremely rare, each occurring in just a few families. Much more common are complex or polygenic disorders, which include many of our most common ills: asthma, schizophrenia, depression, congenital heart disease, hypertension, diabetes, and cancer. These are caused by the interaction of several, perhaps many, genes, each of which alone has only a small effect, and perhaps no detectable effect at all. And typically in polygenic disorders, there's a further complication. These sets of interacting genes may create a predisposition to a particular disease, but whether you actually develop a case of it depends on environmental factors. Suppose you have a set of gene variants that predisposes you to alcoholism. Whether or not you actually become an alcoholic depends on your exposure to the environmental trigger, alcohol. Your fate may be quite different growing up in a dry county in Texas as compared with Manhattan.